All right, hello everyone. We're going to start off chapter 7 with photosynthesis, and I love this cartoon. What do you mean? Oops, this stuff doesn't grow on trees. As you can see, this person has dropped some oxygen off of the boat, and it's a great little comic to start this off. We're going to be talking about how plants intake carbon dioxide, water, and light and produce sugar, and that this thing that we need, oxygen. All right, to start us off, I go over some terms that you should probably already know. Autotrophs, these are organisms that make their own food. We are not autotrophs, even though we sometimes make our own food. It's not the same process. You might heat up a bowl of SpaghettiOs. That doesn't mean you're an autotroph. You are eating other organisms for food. That means you are a heterotroph. Autotrophs are organisms that produce their own food by processes called like photosynthesis or chemiosynthesis. We don't specifically go over this one. I focus more on the photosynthesis, or I should say AP Bio focuses more on the photosynthesis, but they use energy from the sun to make their own food. Heterotrophs, nope, we don't do that. We eat other things for food. Another way of looking at this is autotrophs are producers, heterotrophs are consumers. All right, so taking a look at some of the inputs, I would know this equation, 6CO2 plus 6H2O, and light produces sugar and oxygen. The inputs, water we get from the roots of a plant. Remember that there is a misconception that, the roots come in from, or the water comes in from the leaves. Nope, carbon dioxide comes in through the leaves, and they go through something called stomata, which we're going to get here in a little bit. Light energy from the sun, we're going to talk a lot about this in this chapter, how light is used within these reactions. The outputs, sugar and oxygen. So sugar is used for cellular respiration. It's the main product, and oxygen is the byproduct. And this is where I kind of like to tell students, like, plants don't wake up in the morning and think like, oh, I can't wait to go through photosynthesis to produce oxygen. They really don't care for us. They are producing oxygen as a byproduct. Their main goal is to produce sugar for themselves. They're trying to produce that sugar for cellular respiration, for structural purposes, a lot of different purposes. But basically, this whole process is to make sugar. Oxygen is just a byproduct, and we're going to talk a lot or more about that later on. How do we know that this is a byproduct? We're going to talk about something called the Great Oxidation Event. All right, moving on. So stomata, or stoma for singular. Stomata are openings on the underside of leaves. This is where CO2 enters, and water and oxygen is going to leave, pun intended. But this is where the gas exchange occurs. And I think I put it here. Nope. We'll talk about this again later, but basically these stomata open and close during different points throughout the day. When it's really hot out, the plants will close their stomata to save on water, because water's leaving. If it's too hot, you're gonna have a lot of water loss, and to prevent the plant from dying because it doesn't have enough water, the plant will close these stomata. But that's going to create a problem, because then we can't get CO2 in, and that's a problem for plants, because plants need to take in CO2 to make their glucose. And if they can't do that, they go through something called photorespiration. But we'll get to that later. But basically, again, stomata, these are the openings on the underside of leaves. Factors that influence photosynthesis, I like to talk about light, carbon dioxide, water, and then temperature. Light, carbon dioxide, and water, it will increase to a point. So as we get more light, you get more photosynthetic rates, and you can kind of think about the Earth and where we see more photosynthetic rates. That's like the rainforest and anywhere around the equator, because there's a lot of light present. Places like the North and South Pole won't get a lot of light, so they won't produce a lot of photosynthesis. Same with carbon dioxide. As we increase carbon dioxide levels, we get more photosynthesis, and this is, again, because this is a substrate for photosynthesis, but it will level off like a substrate concentration would. Now, temperature is the graph that we saw for enzyme rates, and this is basically the same reason, is because, too cold, the enzymes can't work, too hot, the enzymes can't work, but right in the middle, it's perfect. So like this would be like the rainforest, and this would be like the deserts over here, where it's just too hot for things to survive. Now, I like to talk about this question, I like to pose this question in class. Why isn't there a large hole around trees when they're like these mighty redwoods? They weigh up to 50,000 pounds or 25 tons. There's a big misconception here that most of the mass of this tree comes from the ground. And you can see this person here standing. This person standing here and showing the relative size of this tree. It's huge. It's humongous. It's there's a lot of matter there. So why isn't there this just big hole around the mighty redwoods? Well, did I go too quick? I usually show a video and I talk about how most of the mass of a tree comes from the carbon dioxide. So going back here, let me go back just for a second here. Going back here, the carbon dioxide that the plant's going to take in, that's going to be the main source of matter for plants, not the nutrients they get from the ground. It's only like 5% of the mass comes from the ground. Okay, moving on here. So photosynthesis, the absorption spectrum. Light is absorbed by molecules called pigments. We're going to talk about that next. But you can see here the reason why pigments, okay, right, why plants are green is because we have these high absorption rates in the purples, blues, and reds, and low absorption rates in the greens here. So you can see this absorption rate where they're not really absorbing greens, they're absorbing a lot of reds, blues, and purples. That means that the plants are going to reflect green. Let me go back here, pigments, there we go. These are molecules that absorb certain wavelengths of light. So you can see here, they're absorbing some, but not all. Chlorophyll is going to be the main pigment that we find in plants, and that's going to do most of the light absorbing. And again, it absorbs a lot of reds, blues, and purples, not greens. That's why plants appear green. I'm going to go through here kind of quickly. Is there anything else? I think this is the end. Yep, this is the end of 7.1. All right, 7.2 is going to focus specifically on chloroplasts. Remember that chloroplasts have two membranes, and that comes from that endosymbiotic theory, where the eukaryotic cells millions and millions of years ago wrapped its own membrane around these guys. These were once living organisms, that they were bacteria that got engulfed through a process known as endocytosis. That was from chapter 5. And the eukaryotic cells wrap that second membrane around these chloroplasts. So there is a couple different parts within the chloroplast we have to know. Stroma is the solution inside of the chloroplast. It's like the cytoplasm of the chloroplast. This is specifically where the Calvin cycle is going to occur. We're going to be learning about the light independent and light dependent reactions. The Calvin cycle is the light independent reactions. These are going to go in, again, the stroma, the liquid, the cytoplasm of the chloroplast. A couple other structures. So we have these photosynthetic discs that I like to call them. They're called thylakoids. A stack of them is called a granum. Grana for plural. There's all these words. And then the lumen is the liquid inside the thylakoid. We have to specifically know each of these locations. I talk about this in regular biology and like, hey, you know, these really small structures within the chloroplast. We have to know where these are located because this is the first step to learning the light independent and dependent reactions. Think about what these thylakoids do. We talked about a really important concept within cell organelles. What do these thylakoids do for chloroplast? Think about it. It's a long word. Starts with a C. They compartmentalize the chloroplast. So we can have specific structures going on within these thylakoids. Here's what a chloroplast actually looks like. So again, I love this. This is the David Goodsell image. You can see the thylakoids. They are just photosynthetic discs. I like to call them photosynthetic pancakes because I think they look like pancakes. But anyways, but this is what they actually look like. So a little bit more in depth, but this is what a chloroplast looks like. Now, within the chloroplasts are going to be something called photosystems. These are a group of pigment and electron acceptors. These are going to be specific to the light. Let me go back. Light dependent reactions. This is just basically a solar power plant, and each one of these circles here represents a pigment. So we talked about pigments in the first section. These pigments are going to absorb light energy, and they're going to transfer it to things called electrons that are from water. And I'm going to get more in detail with that, so don't get overwhelmed with that. But there are two different types of photosystems that we see in the light dependent reactions, and they do have different structures, which means they do have different functions. So you can see their structures are different, and we're going to be going over these here in a little bit. I think that's it. Let me see. Yeah. Oh, you know what? I'll just talk about this here first. So before I let you go for 7.2, there are two specific reactions. Like I said, these are going to be really complicated, but it's really important to remember that these are almost like a story, and you have to kind of memorize this story. We have the light-dependent reactions and the light-independent reactions. Light-dependent reactions require light. Light-independent reactions don't require light. The light-dependent reactions occur in the thylakoid, specifically the thylakoid membrane. And they only occur when there is solar energy or some type of light available. Solar energy is used to produce ATP and NADPH. We talked about ATP before, not NADPH specifically. We'll get more in depth with that again in the next section. The Calvin cycle is the light-independent reactions because, again, you do not need light for this reaction. This occurs in the stroma of the chloroplast, and this is a process where we're going to take carbon dioxide and actually make carbon dioxide, and we're going to make carbohydrates in the form of sugar. Chemical energy is in the form of ATP and NADPH is used to produce these carbohydrates. Yeah. So we're going to be learning the similarities and differences about these two different processes. All right, so now time to dive into 7.3, which is going to specifically talk about the light-dependent reactions. I like to show this graphic before getting into both of these reactions. We're going to look specifically right now at the light reactions. We are going to see that these light reactions are going to power the Calvin cycle. The Calvin cycle occurs in the stroma, while the light reactions, again, occur in the thylakoids. There are going to be two molecules that are going to be exchanged here, NADPH and ATP. In the light reactions, we're producing oxygen from the breakdown of water. From the Calvin cycle, we're producing, it says CH2O, and we're producing that from carbon dioxide. And these CH2Os, that's just showing us a carbohydrate in some form. So we're producing carbohydrates from CO2. And we're also having this exchange of energy between these two cycles, between these two molecules. All right, now, a lot of words there. Let's get into the details. Before we get into the specific steps, let's talk about NADP. This is a coenzyme. It's going to accept and deliver electrons and hydrogen ions between the processes. There is a reduction in oxidation reaction that occurs here. So when NADP gains the electron, it's going to be reduced. When it drops them off, it's going to be oxidized. Remember, oxidation is losing, reduction is gaining, or oil rig. I'm not going to show this video, but I'm just going to talk about the steps here that are involved in photosynthesis and specifically the light-dependent reactions. All right, so water splitting. This is the first part of the light-dependent reactions. Water is going to split. It's called photolysis or photolysis. This donates electrons to photosystem 2. Oxygen is going to be left over, and this is where the oxygen is going to be produced. Again, this is a byproduct. Hydrogen ions create a gradient for ATP. Light is absorbed by pigments in photosystem 2. That energy is going to, from the light, is going to be transferred to the electron. They're going to become more energized here in the PS2. Now, there's a lot of words there. Let me show you what that looks like here. So we have our chloroplast stroma, we have our thylakoid lumen. Water is going to break down. It's going to donate its electrons to photosystem 2. Photosystem 2 is going to energize these electrons by absorbing light through these pigments. Now, when water breaks down, it's going to break down into oxygen and hydrogen ions. Oxygen is going to leave through the plant through diffusion, and this is what we breathe in. Hydrogen ions are going to create a gradient here with a high concentration in the thylakoid lumen compared with the stroma. So this is the first part of the light-dependent reactions. We have energized electrons, we have hydrogen ions, we have oxygen. Yeah. So the reason why we do photosystem 2 instead of 1, scientists, when they were discovering the different photosystems in photosynthesis, they discovered 1, and then they discovered 2, and then they realized later on that 2 occurs before 1, and then they never changed it back. So the joke there is, why didn't they change it back? They just didn't, and they should have, because it can be confusing. A great reminder that hydrogen ions are just protons. You can see this hydrogen atoms, hydrogen ions have lost that electron, so it's just a proton. All right, the second part of the light-dependent reaction. This is where we're going to go down the electron transport chain. Electrons move down this ETC, and what's going to happen is that these membrane proteins are going to use the electron's energy and actually force more hydrogen ions into the lumen. So we're creating an even higher concentration of hydrogen ions. Photosystem 1 is going to accept these electrons from the electron transport chain. The photosystem 1 is different. We talked about the structure is different. They are still going to energize the electrons by using light and absorbing that light and then transferring it to the electrons. But basically, it does do different, like it's different structurally, so it's going to have a different function. We don't break down water here. The electrons then are passed to an enzyme in the membrane called NADP reductase. This is going to bind those electrons plus a hydrogen ion to make NADPH. So the final electron acceptor in this process is NADPH. Let me show you what that looks like, because this is just a lot of words. So again, electrons are fed into the photosystem 1. They're going to become re-energized. They are going to, these re-energized electrons, they are going to go to NADP reductase. They are going to bind with NADP and a hydrogen to form NADPH. You can see these two electrons here, these blue circles, they are going to go to the next reaction. So our first main product here of the light-dependent reactions is NADPH. All right, so now that we have the light-dependent reactions done, we're going to get into the light-independent reaction. These are a series of specific reactions that occur within the stroma, and they're going to occur after the light reactions. Now, we used to call these the dark reactions, but there's a misconception when you call it a dark reaction because you think it occurs only in the dark, because the light reactions only occur in the light. But these reactions occur both during the day and the night. So it doesn't need light, or it doesn't not need the presence of light, if that makes sense, but it just occurs without light. There are three specific parts here. We have carbon fixation, carbon reduction, and the regeneration of RUBP. Here is the process. Don't get too overwhelmed. You have to focus on the carbon. So these little purple circles are carbon atoms, and we are going to focus specifically on those carbon atoms. So let's talk about the first one. Carbon fixation. This is a cycle. So again, think about enzymes. Carbon fixation occurs when a single molecule of carbon dioxide attaches itself to a 5-carbon molecule called RUBP. So you can see RUBP has 5 carbons. CO2 has 1, and the enzyme that does this is called ribisco. Now, this ribisco actually has a longer name. You might think, Mr. Frat, if it doesn't have an ACE at the end. This stands for ribulose 1,5-biphosphate carboxylase. Yeah. So we just call it ribisco because that's a really long name to remember. The 6-carbon molecule then splits into two 3-carbon molecules called 3-PGA. So again, 5 carbons, 1 carbon, 6 carbons. 5 plus 1 is 6. This instantly breaks down to 2 of these 3-carbon molecules called 3-PGA. That's carbon fixation right there. Boom. This is the first part of the Calvin cycle. Now, interestingly, a lot of people think that the carbon is attached to the end, but it's actually attached to the middle here. Not that you need to know this, but this is what that reaction looks like. Somebody asked me about that once. They said, where is that carbon atom actually attached? And I said, I don't know. I think it's at the end. And I learned it's not the end. It's actually in the middle. Ribisco is the most abundant enzyme on Earth. It is the enzyme that, again, fixes carbon into RUBP. So it's the most abundant enzyme on Earth because there's a lot of plants, and it's a very slow-moving enzyme. Carbon reduction is the next part here. What's going to happen is ATP and NADPH are going to reduce the molecules in this reaction, and they come from the light-dependent reactions. What happens to the 3-PGA is it's reduced into G3P, which is another 3-carbon molecule. You can see that these 3-carbon molecules and these 3-carbon molecules look very similar. Again, their structure is different, but just understand they get changed, and they just remain. They don't. The carbons remain the same. They don't lose any carbons. Last step here, this is the regeneration of RUBP. So these G3P molecules are actually really versatile. We can make these G3Ps into a lot of different molecules. What happens in this part is you are going to regenerate these G3Ps into RUBPs. Now remember, the RUBPs have 5 carbons. These only have 3. So you can see these numbers in the front here. They're going to represent that we have to do this with multiple molecules. And again, there's a lot of enzymes that work here. We don't need to know those enzymes, but just remember that ATP powers those enzymes, and that comes from the light-dependent reactions. So this is what I initially kind of show you. Just doing the math, you have to kind of triple everything. So this is more what it looks like. So you can see the 3 becomes 6, 6 become the 6 here, 5 go on to regenerate RUBP, and the cycle continues. However, these 3 carbon atoms that come through this process in the form of carbon dioxide are made into 1 G3P molecule. Now you might think, hey, this glucose has 6 carbons. Why does this one only have 3? Well, you have to essentially double that because the reaction will have to occur twice to make 1 glucose molecule. So it's a lot to take in, but these are the light independent reactions. So 2 of those G3Ps will make 1 glucose molecule. This whole process, like I said, occurs twice. Now, G3P can be made into a host of different structures. It can be made into glucose. They can be made into fatty acids, amino acids, fructose, a lot of different molecules, starches. So they can be made into a ton of different things. So this is that starting molecule that plants are going to use to make their carbohydrates. I think that's it. Is there anything else we're going to talk about specifically? I talk about a little bit about the evolution of this process, but not greatly in depth. Yeah, this is the end of 7.5. All right, so this process right here is showing what we just talked about, or this picture is showing us what we just talked about. So we have light being absorbed by photosystem 2. It's gaining electrons from the water. Water's dumping off hydrogen ions in the lumen here. We're creating this high concentration here in the lumen, and this is also producing our byproduct water. byproduct oxygen. The electrons are going to move down this electron transport chain. They're going to force more hydrogen ions into the lumen here. You can see the electrons being re-energized here at photosystem 1. Photosystem 1 then dumps those electrons off onto NADP and a hydrogen ion and forms NADPH. So again, this whole process is basically producing a high concentration of hydrogen ions here in the lumen. What we're going to talk about next is the ATP synthase production, or ATP is being produced. So we have this high concentration of hydrogen ions here compared with outside in the stroma. So what's going to happen is these hydrogen ions are going to want to move from high concentration to low concentration. And what happens is the hydrogen ions move through the ATP synthase and actually make it rotate. And this rotation is going to force a phosphate group onto a DP, making it a TP. Remember, adenosine diphosphate has 2 phosphate groups. Adenosine triphosphate has 3 phosphate groups. Hydrogen ions move through ATP synthase. Good. So this is specifically known as chemiosmosis. This is the movement of ions down a chemical gradient, and its final product here is ATP. So we're trying to produce 2 things in the light-dependent reactions, ATP and NADPH. Here is that enzyme working. I love the look of this. So this is ATP nested mitochondrial. We will see these in the mitochondria as well, but basically you can see this ATP synthase rotating. It just looks beautiful to me. This is what it actually looks like. It's a protein, so you can see those protein structures there. Here it is again. And what you can see here on this right-hand side is the phosphate groups being bonded onto the ADP molecules. These ADP molecules are low energy molecules. The ATP molecules are high energy molecules. I'm not going to go over these 2 videos, but here again is that process. We talked about this process a little bit before, but again, our hydrogen ion concentration is going to be high in the lumen and low in the stroma. It's going to move from high to low concentration, so it's going to move through here. It's going to rotate this, making ATP. So our 2 products here are ATP and NADPH. Here's another great animation. If you follow me on TikTok, you might see me post this. I talk about what's going on here. So again, water's being broken down, donating electrons. Hydrogen ions are creating a high concentration here. You can see the photosystem 2 is going to energize the electrons, which move down the electron transport chain. They are pumping more hydrogen ions into the lumen. We then re-energize those electrons. They go into make NADPH, which is a product. The hydrogen ions move through ATP synthase, making ATP because of that rotation. And that is what you got to know for 7.3. All right, our last section here in photosynthesis is going to talk about differences within photosynthesis and other plants. So we have C3 plants. This is the majority of plants that carry out photosynthesis. They carry it out just like I described in the past couple sections. They occur in most environments. They call them C3 plants because the first molecule that's produced is that 3-carbon 3-PGA. What's going to happen, like I talked about, is these molecules or these stomata are going to allow CO2 to enter, but they're not going to allow, hold up, let me back up. They're going to allow CO2 to enter, and they allow water and oxygen to leave, like we talked about. However, during hot portions of the day, they're going to close up because it's too hot and we don't want to let water out. However, that's a problem because because when the plant doesn't have enough carbon dioxide, it's going to have low levels of carbon dioxide and high levels of oxygen. That's because, again, the stomata are closed. What's going to happen is Rubisco is going to take up oxygen instead of CO2 because there's no CO2 left. And that is a problem. This is not good for a plant. You can see Rubisco here. This is called photorespiration, when Rubisco takes up oxygen when there's low levels of CO2 compared with high levels of CO2 when we have the Calvin cycle occurring. This is called photorespiration. And I love this. Rubisco usually binds with CO2, but sometimes it will bond with oxygen when there's high levels of oxygen. What happens during photorespiration is instead of taking in CO2, the plant is going to respire CO2 or let out CO2. And that's not good for a plant because when plants let out CO2, they're not taking it in, which means they're not taking in carbon to make glucose. And when they're not taking in carbon to make glucose, they can't make glucose. And if they can't make glucose, bad news bears. They're going to not do so well because they're not making food for themselves. So you can see the normal Calvin cycle, we're taking in CO2 when producing glucose. In photorespiration, we're taking in oxygen and we're producing CO2, and that's not good because we're letting out CO2. Now, why does this occur? We talked about in enzymes, because Rubisco is an enzyme. Why does Rubisco take up both CO2 and oxygen? I thought enzymes were supposed to be specific. Well, remember, this process evolved when there was very small amounts of oxygen on Earth. And since there was small amounts of oxygen, Rubisco didn't need to be specific to oxygen or CO2. It was just grabbing what it could. And it's not specific to either. So it's a problem that plants have because of the atmosphere at the time. So how have plants figured out a way around this? We have C4 plants, and these live in hot and dry environments. The first molecule they make is a 4-carbon molecule. That's why they call it C4, not because of the explosion, but because the first molecule is a carbon 4-carbon molecule. So what happens here is in these C4 plants, they don't take up oxygen because they have a different enzyme. It's called PEP carboxylase. PEP carboxylase can't use oxygen, so that's how they get around. Even if they do have high amounts of oxygen, they won't pick, or the enzyme won't pick it up. Now this costs a lot more energy, or it's not as energy efficient, but it's much more effective in these hotter environments. So you can see here this PEP carboxylase, it's going to take up CO2 and it will not take up oxygen. It will make this 4-carbon oxyl acetate molecule, which will produce CO2 for the Calvin cycle, and it protects this Calvin cycle from taking up oxygen in photorespiring. You can see the difference between the C4 and the C3 plants. You can see that the C4 plants are mainly around the equator. Cam plants are a different type of plant that, again, live in hot and dry environments. They only open their stomata during the night, and this allows them to take as much CO2 as they can during the night so they can have CO2 throughout the day. And again, cam plants will look a little bit different than C4 plants. They will still create a 4-carbon molecule, but they'll do this just a little bit differently than the C4 plants. But basically what I'm just talking about is these plants evolve different ways to, in response to those stomata closing. Yeah, I love this meme. So C4 or cam plants and C4 plants, they don't go through photorespiration because they've evolved processes where they don't need to. They still go through photosynthesis. Good meme. All right, so this is the end of the chapter. Yeah, this is not the worst of it. This is just the half of it. We're going to see cellular respiration next, and it's a tough chapter, but this is the end of chapter 7.